Hello, everyone. Thank you so much for joining us. My name is Wendy Xie, and I'm a project manager at the National Collaborating Center for Infectious Diseases. I would like to welcome you to this webinar where we will explore the question, what can routine surveillance data reveal about antibiotic resistance dynamics with Dr. Gwen Knight. I would first like to acknowledge that the National Collaborating Center for Infectious Diseases, or NCCID, is hosted at the University of Manitoba, which is situated on the ancestral lands of the Anishinaabeg, Anishinaabeg, Dakota, Dene, Nehithawak, and Ininiwak nations. It is also in the heart of the Métis Nation. I am joining this webinar from the unceded lands of the Anishinaabe Algonquin Nation. At NCCID, we strive to honor the lands and their original caretakers in our work, and we acknowledge that we are on Treaty 1 land and recognize that this and other treaties have been implemented as a part of the process of colonization intended to benefit some while harming others. We are committed to working with our partners towards reconciliation. So just a few housekeeping items before we begin. We'll be using the chat tab for communication with attendees, so please check there for updates. If you have any technical problems with Zoom, the chat tab has been disabled for participants, so please send us an email at nccid@umanitoba.ca. If you have questions for the speaker during the presentation, please leave them in the Q&A tab, and we will try to get through as many as possible after the presentation. And just a reminder that you can give a question a thumbs up in the tab to push it up in priority. And finally, the recording of this webinar, along with a copy of the presentation slides, will be available on the NCCID website under the webcasts tab soon after the event. And now I would like to introduce our speaker for today. Joining us all the way from the UK is Dr. Gwen Knight, who is an associate professor at the London School of Hygiene and Tropical Medicine, or LSHTM. Gwen is also co-director of the LSHTM Antimicrobial Resistance Center, and her research is focused on math modeling of the dynamics of antimicrobial resistance, or AMR. She currently has projects exploring the interaction between demography and AMR, One Health approaches to AMR at the farm level, and the spread of resistance genes by bacteriophage. So thank you so much, Gwen. You have the floor.
Thank you, Wendy. And can you see my slides? Can you hear me?
Yes, thank you.
Great. Well, hello, everybody. And thank you to the organizers for giving me the opportunity to speak today. I have to say, I'm very excited to be here mainly for three reasons. So the first reason is, as Wendy said, I'm sitting in London, and I believe many of you are sitting in Canada. It's one of the few, obviously, great things about COVID that these kind of events can happen, do happen, and I really want to say that we should reach out. I think antibiotic resistance, as many of us know, is such a global public health problem. I really want us to be a global public health community, and please do sign up to our London School AMR Centre newsletter. We have webinars. We'd be very welcome to you to join our webinars too, and do contact me after this. Very open to collaboration. I think we should try and make the world a smaller place without burning up our carbon footprint and actually traveling in person. So that's my first point. So thank you for welcoming me today. The second point is that from conversations with Wendy and colleagues, I'm very excited that the audience today, I think, is quite diverse. So it's not just researchers, it's some of you are really at the AMR or the antibiotic resistance coalface, working with patients and prescribing antibiotics. And although I'm a mathematician, I hope that I can show you how useful mathematicians can be and modelers can be, and hopefully I can talk to you a bit about the importance of the data that we're collecting at that coalface and how we can use that to do better research and design better interventions to control this global public health problem. And thirdly, I'm very excited to be here because I really like speaking about antibiotic resistance. I think it's, as I said already multiple times, a huge global public health problem, and we need to bring multiple disciplines together. And so I would describe myself as a mathematical modeler, but I really like this image here and this idea that mathematical models are like campfires. So I hope what I can do today is to put some ideas out there, and I can put down my model of how I think the world works and how I think I should build my mathematical model of the spread and the dynamics of antibiotic resistance. But I really want to see that as like, if I write this down, please come and destroy it. Come around this campfire and come and tell me where I'm wrong. Come and tell me where I'm right. Come and tell me where there's extra evidence that I'm not citing and that I'm not using and that I should be using or I should be challenging. And really trying to provoke increased and better research going forward in the field of antibiotic resistance. So I'm hoping that many of you are familiar with antibiotic resistance. I won't go into a lot of detail about it, what it is, but I wanted to emphasize here that I am a modeler who mostly thinks about bacteria. So I mostly think about antibiotic resistance, which is a subset of antimicrobial resistance. And it's very simply a definition, when bacteria survive in the presence of an antibiotic, it's said to be resistant. So here, bacteria going on a plate, and each of those little circles are impregnated with antibiotics, and how far that circle of clear growth, where no antibiotic, that bacterial lawn, where none of it grows, the size of that diameter tells you how resistant that bacteria are. And so this is obviously a problem, because when bacteria cause infections and those bacteria are resistant, we have increased mortality and morbidity because we're unable to treat those patients. And so I really want to center my questions and my research around trying to prevent infections with drug-resistant bacteria, and how we can really face that acute final problem, reduce the number of drug-resistant infections. So as I mentioned, I'm a mathematical modeler, and for those of you that aren't mathematical modelers, I often, when I present to non-modeling audiences, there's often a bit of skepticism. What's the model going to do for us? I hope, and many of us in the modeling field are very happy that models were used so much during COVID. I hope you have a much better awareness of the power of models. But I just want to briefly again say here why I think building models is so useful. So fundamentally, I think we all have a model of how the world works in our head. We will have a model of, if you look at the sky and there's dark clouds, it's probably going to rain. We have models of the way the world works, and we're not questioning them. We're not writing them down. We're often not challenging them. And if we write down a mathematical model of how something like antibiotic resistance spreads, we're putting, as I said, we're putting something there that can be challenged, that can be explored, that can be parameterized, that can be checked, and can be used to do research and to try and improve our understanding of how the world works. So here I have a very simple image of the iterative process that I believe should always happen in research, but particularly in mathematical modeling, where you have at the top this sort of standard natural history diagram where you think of individuals as not carrying an infectious disease, being susceptible, and then getting infected at a certain rate, here beta, and then moving to becoming infected, and then moving to becoming recovered after some period of infectious NAS. So this is a sort of standard SIR model that you might have heard of before. And here I'm using that as an example. I'll talk a bit about antibiotic resistance models in a minute, but that is a simple representation of what I would call a natural history or population dynamics model. We can also think of statistical models, which I'll talk a bit about today, or we could think about things like time series models. All these models can be written down in the next step. Once you've built your model, written them down in some kind of mathematical, black and white, logical, technical language that again can be very clearly, and we've got lots of tools that we can use to explore this mathematical language. So in doing that, there's already a lot of understanding that we bring. So we're already saying, well, what are the key populations we need to include? What are the key rates? Is there immunity? Is there not immunity? Do we need to think about subsetting populations by different risk factors? We're already, by building that model, we're already proposing and gaining new understanding of how the system works. And then, the part of it, once you've written your model, is checking that it represents accurately what you want to represent in the world. So this is where I would say comparing it to data. So can we capture, for example, the size of an outbreak, the peak of an outbreak, the number of people that will get infected or become immune after a certain period of time? I'm not going to talk about it today, but as Wendy mentioned, part of my research is looking at phage dynamics and working with microbiologists to understand competition and fitness costs. And when we do that, there are many things in the lab that we can't measure. For example, phage that are carrying non-phage DNA, like carrying bacterial DNA. We can't actually measure them very easily in the lab. And so we can build a model to capture some of the dynamics, so certain populations of bacteria and phage, and we can use the model to reveal hidden dynamics. And that was very powerful during COVID, for example, to reveal things like the level of asymptomatic transmission, which was very hard to measure from the data. So once you've gone around this circle a few times, you've thought about your model, you've spoken into disciplinary meetings, you've brought people around your campfire, you've written it down in the logical language, you've compared it to data, you've probably changed your model, you've tried to estimate some of the parameters, then you've really got a tool, and that's really powerful. So you've got this tool, this mathematical modeling tool that you can use to generate things like burden, like how many people have got certain infections or in a certain state, and then you can use that those burden estimates to estimate how many people will have it with or without certain interventions. And then obviously with quantifying the impact of interventions, you can then go on to try and make better decisions, better evidence-based decisions, obviously conveying within that all of your uncertainty about model structure, about data, about parameters, right the way through to potential impact of interventions. So that to me, those to me are the reasons why I do modeling. I do modeling to do all of these things, to try and build understanding, to try and estimate impact of interventions, and ultimately to support decision making in the AMR space. So that was the model process and the SIR model. Now I'm going to talk about what does a model of antibiotic resistance look like. So it's slightly different in that we have to think about non-colonized individuals, as in susceptibles is a very nice common word used in other infectious disease dynamics, but obviously that hits with our definition of resistance and has a very different meaning. So if we think about many bacterial pathogens, then we have a state where there's non-colonized, and we might actually think for many bacteria that there's not very many people in this state, but we have individuals that are not carrying this bacteria, and then some of them are carrying or colonized with the susceptible pathogen, and some of them are colonized with the resistant pathogen. And the key thing here is that this is the asymptomatic state, so these are not infectious. Well, okay, so they could be infectious, but they're not infected, they're not symptomatic. And so there's some kind of progression from colonized to infected. And then obviously once you're infected, we assume that there's some chance that you'll be exposed to antibiotics, some kind of treatment. And we assume that that treatment would be more effective for the susceptible pathogen than the resistant pathogen. We also assume, and this is something that I'll come back to later, we also often assume that if you're exposed to antibiotics, then you're more likely to move from being infected with the susceptible pathogen to being infected with the resistant pathogen. So there's some kind of selection event here where you select for maybe subpopulations within that infection that are resistant, or you clear the susceptible carriage and allow for resistant bacteria to successfully transmit to that patient. So there's some movement linked to antibiotic exposure. Now, this is a very simple model. I'm sure you're all looking at this thinking, what about this arrow? What about this complexity? A few questions that this originally raises for me. Well, what rate do patients get exposed to antibiotics? Or what rate do they get exposed to antibiotics that actually would treat a resistant infection? And what about mixed carriage? Can you carry both susceptible and resistant pathogen? Many different complexities that can be layered on top of this very simple natural history model of resistant pathogens. The main complexity I want to expand on now is this one over here, and that's the idea that we think about antibiotic resistance as being you've either got it or you haven't, but actually antibiotic resistance is highly complex because it's not just resistant to one antibiotic, there are many antibiotics in the world, and so actually we need to think about potentially a model that looks like this. So individuals not carrying any pathogen or colonized with the pan-susceptible pathogen, and then colonized with all the different combinations of susceptible and resistant to the many antibiotics that we know are out there, or maybe the three that are most important for this pathogen to use clinically. And then the same idea that you're likely to be asymptomatically colonized before you progress to infection. So this is likely to vary by the bug. Some bugs are more likely to have a long asymptomatic state before progression to infection, or you might believe that that's really short. And actually, we should blend those two states together. And then if you're colonized, obviously you can become infected with all of those. And then this is where it starts getting a little bit crazy, because the number of parameters and the number of rates and the number of transitions just starts multiplying up by all these combinatorics. So you've got how effective is treatment for each of these different levels of resistance. We've got that movement from pan-susceptible to each of those, probably one first, then two, or maybe if they're on the same genetic element, it moves quicker, huge amount of complexity. And then the movement between these antibiogram combinations, so these combinations of resistance, the gain and the loss of resistance. At what rate does that happen? What rate does that happen during infection? Or what rate does that happen during colonization? What's the impact of these resistances on treatment? As soon as you start writing down even the most simple mathematical model for AMR, it becomes really, really complicated. Like all good science, the way to cut through that is to come up with your key question. What is your pathogen? What is your drug resistance? What is your setting? And that can help to narrow down what you need to build into your mathematical model. But I just want to emphasize here that even thinking very simply, mathematical models of AMR can get very, very complicated. Now we're at that point, as I showed you on that iterative diagram, of saying, well, I've got my model where I could write down some equations. I need to now compare it to some data. Now, this is where I hit a huge problem in the antibiotic resistance field, and that's really what I want to talk to you today, is this problem about the data that we have in the field of AMR, and why, as a quantitative scientist is trying to build models of antibiotic resistance, it's very not only troubling, but I think worrying in terms of what we do with that data and what we understand about that data. So what data on antibiotic resistance actually is available? Now, if we were to think in an ideal world, what data we want on antibiotic resistance, then we would want to sample everybody quite often, maybe every day. We'd want to sample from places where we'd find bacteria, so skin, nose, groin. You'd probably want to sample fecal matter, maybe urine as well, maybe multiple points on the skin. We want to know what antibiotics they're exposed to, but we want to know where they go. Do they go to hospital? Do they go to a nursing home? Do they go to a pharmacy? We probably want to know when they go to hospital, what they get infected with, what their symptoms are like, what they're exposed to, where their infection is, whether it's on the skin, starts as a wound infection, then progresses to bloodstream. We want to know the resistance profiles of all of those colonizing and all of those infecting bacteria. And we'd want to know the diversity within a patient and how the list becomes huge. And I used to be someone that wanted to do those studies, that wanted to collect all of that data. And I've moved more into the fact that that's just not going to happen. It's not going to happen globally. It's not going to happen routinely. It's not going to happen in any level of detail. Or we'd also want to sequence, of course. And instead, the data that we have on antibiotic resistance, the data that we are going to compare our models to, these statistical simulation population dynamic models, the ones that we're going to be able to actually really check what our models do, partly it's going to be lab data. So part of it is going to be trying to exploit in vitro conditions where we have strains, where we can put them together, we can look at growth. Some of it is going to be from that. But the main source that we're going to get is from routine surveillance data from patients with infections in hospitals. That's going to be the main data that we have on AMR, because we're not going to sample patients when they're asymptomatic. We're probably not going to sample patients in the community either. But we do have data from patients when they got an infection. Often we have data from a patient or a sample from a patient before they're given an antibiotic. There are, of course, going to be one-off prevalence surveys, so sampling in a certain community, gathering of E. coli from fecal matter from a certain subset of the population. We are going to have snapshots like that. We're also going to have snapshots of that kind of longitudinal data, and they are going to be very important for our modelling. But I think it's really important to emphasize that so much of what we know about AMR in terms of our burden, our impact of interventions, is based on this routine surveillance data. And if we think about going to back to this model, what part of this model is that data from? It's really from this one subset here, this one population here. And I've deliberately emphasized the infections with the triple resistance here, because if an antibiotic is working, antibiotics are extremely cheap. We often give them empirically without checking what the microbiological diagnosis is. And that's totally fine. That's totally linked to the cost effectiveness, the time, the diagnostic tools that we have right now, that's another aspect of this data, that it's patients with infections that we can sample from, where the empiric standard therapy is not working. And so that's likely to be the very tip of the iceberg, those patients with the most resistant infections. So does that matter? Because, okay, yes, if I want to build my complicated model, I'm going to struggle to find some of these parameters, but maybe that, maybe that's okay. Maybe I can work these things out in a different way. Maybe I can use point prevalence surveys or lab data. Maybe that's okay. I think what worries me is that we use this data for a huge amount of things in the AMR field. For example, coming up with national level estimates of resistance to drug X in drug X in bug Y in country Z. And that misses a huge amount of information that is based on, as I said, this tip of the iceberg. It's based on, for example, E. coli isolates from bloodstream infections, and from those that are not that are failing empiric therapy. Coverage will sometimes be better in, for example, high-income countries and lower middle-income countries. We know very few of even the most serious infections are sampled. And why does this matter? Why does it matter? We've got an idea of in the most acute, most worrying infections, what the level of drug resistance is. Maybe that is what we need to know, because that's where the most worrying thing is. I think it's not where I think we need to know a hell of a lot more. We need to know all about drug resistance dynamics in the asymptomatic population. We need to know if it's selection or transmission if we want to do something about it. But it also worries me because it means that we're going to get inaccurate burden estimates. It's going to really depend on which patients are sampled when and why. We might also get inflated concerns about resistance prevalence if we are only taking the tip of the iceberg, and if a certain country, that denominator is really reduced, it's going to push up resistance prevalence and look like resistance is much more of a problem. Or maybe resistance to a first-line drug that is commonly tested for is a big problem, and therefore prescribing recommendations move on to prescribing second and third-line drugs, when actually there's a bias in the surveillance that means that resistance is not actually that wide in the general population. That leads us to poorly targeted interventions and potentially missed opportunities to understand AMR drivers. And I'm going to talk a bit about that, poorly targeted interventions and understanding of drivers at the end of my talk, when I go on to some of my latest research on demographic variation. So I've told you how bad it is, and I've also told you that I'm not a convert to this, like, go out and sample everything and sample everyone every day. And that's because, as I said, most of our data is routine surveillance data. And so as someone that was getting concerned about biases in the denominators and getting concerned about, well, is it really okay? Like, how bad is routine surveillance data? How much does it change over time? What are the limitations and how far can we push it? So I'm now going to talk to you about two pieces of research that I've done. So the first one was focused on a single hospital, where I really wanted to say, okay, if we just look at this hospital, what happened over time to routine surveillance data? What can we do with this data? How good is it? And then I'm going to talk, as I said, some of this demographic variation, which again uses routine surveillance data, but really shows us something about how we should be thinking about AMR dynamics and AMR variation. So in order to answer this question, how can we use routine surveillance data? How bad is it? How good is it? I was very fortunate to have a collaborator at the Great Ormond Street Hospital for Children, which is a large specialist hospital for children just around the corner from us here in the London School. And they have the most amazing resource, which is Drive, which is a data research initiative, basically. And within that, they have unified both antibiotic usage and microbiology data with patient data to triangulate them into a database that is actually really rare to find, even in the UK with the NHS. I'm really struggling to find these unified databases where you can understand both the pathway of the patient and their antibiotic exposure, and then the linked microbiology. So very fortunate and very grateful to Gosh for setting up this thing, and obviously for all the patients and the samples collected from them. Now, this is work done by my former PhD student, Quentin Leclerc, who's based now at the Institut Pasteur, and his PhD was all about the bacterium Staphylococcus aureus. So this is commensal bacterium. Around a third of us will carry it up our nose, armpits, or our groin, and you've probably heard of it. And I don't actually know what the Canadian acronym is, but it's methicillin-resistant Staphylococcus aureus is the resistant subpopulation of Staph aureus, so MRSA or MRSA. And the problem with MRSA is that once it becomes resistant to methicillin, that's like an indicator that allows it to gain resistance to many other antibiotics. And it's a huge global problem in terms of mortality and morbidity, and causing infections that are increasingly hard to treat. Now, the main way that Staph aureus gains resistant genes is by bacteriophage. So these are bacterial viruses that can kill bacteria, and they can effectively sleep within the bacterial genome. And Staph aureus genomes have a huge number of phage. But they can also spread resistance genes. They can accidentally package bacterial DNA and spread resistance between strains of Staph aureus. And so Quentin's PhD was trying to quantify this rate of what's called generalized transduction and explore that. And when we were talking, and we were doing he was doing lab experience in the lab of Jodie Lindsay, who's based at St George's, University of London in South London. We were talking about this, and then we were sort of saying, well, this is all well and good measuring it in the lab. Is there any way that we can try and measure rates of resistance gene transfer in patients? And this is where we built up this collaboration with Gosh, and we were using it as a sort of two-edged sword. So we were using it to try and answer these questions about rates of resistance gene transfer, and then also for me to try and answer, well, how bad or good is routine surveillance data? So we were looking for two different things. We're basically looking at the hospital level to look at what we could see was happening at the hospital from routine surveillance data, and we were looking at the patient level. And to simplify down a lot of these words here, I wanted to know, what can we do with routine surveillance data? And Quentin wanted to know, well, we both wanted to know, but can we see resistance, can we see diversity and resistance movement at the patient level? So we, the data that we managed to get from Great Ormond Street was, as I said, really amazing. 20 years of data from 22,000 patients. So anyone who was ever positive for Staph aureus, I should note here again that yes, these were children. So most of them were 16 years and younger. And we had some information, the phenotypic information from all of these Staph aureus isolates. Should say here that we didn't know whether it was colonizing or infecting. And we also had linked antibiotic usage and hospital admission data. So I'm just going to walk you through what we found at the hospital level. So at the hospital level, you can see at the top here is the proportion of these Staph aureus isolates that were MSSA, so these highly susceptible Staph aureus strains, or MRSA, so that's shown in yellow at the top there. And you can see that there was an increase in this highly resistant subpopulation starting at around 2014. And from discussions with our clinical collaborators, what they mentioned was that they were concerned that there was an increase in MRSA, and they thought that it might be linked to actually a change in the patient population. And so we looked at the ethnicity of the patients, and obviously we can't prove any causality, but it did align with a decrease in the proportion of patients that were white British. And also, obviously, the COVID-19 lockdown shown in the second vertical dashed line there. The other thing that we asked about was, well, maybe this change in MRSA prevalence was linked to a change in the bugs. Maybe the bacteria changing, there was a new variant or a new lineage in place. And that's happened before in the UK population. But actually what we found was that in 2014, when this change happened, there wasn't really a change in the composition of the bacteria. So here what I'm showing you are the results of lineage typing for a CC. So that's the clonal complex of Staph aureus. It's the way of defining different families of Staph aureus. And you can see from this that what changed was actually in 2011 that there was a change in the surveillance guidance, so change which isolates and what isolates were collected and how they were typed. And so we believe from this data that the increase in MRSA was not linked to a new clone coming in and sweeping, but instead, if you took the data at face value, then you'd think that there was a big change. For example, CC5 strains started to appear in the population of CC8, but actually what happened was in 2011, there was a big change in testing strategies. And it turned out that 2011 was quite a key year for Great Ormond Street, a key year in interpreting data, because a huge number of denominators changed. What I'm showing you here, the proportion of isolates tested for resistance to four important antibiotics, particularly vancomycin, the bottom right-hand side here. And what you can see is that the proportion of isolates tested really dropped off in 2011. And we were told the reason for that was like, well, we weren't finding much resistance, so we stopped testing. Obviously, you're not going to find if you don't test. And so interpreting the trends in vancomycin resistance across Great Ormond Street over the last 20 years would obviously be massively flawed and massively affected by this change in denominator. What we did find, though, was quite an interesting thing in terms of fluoroquinolone resistance. So this is really the proportion of isolates tested that were resistant to ciprofloxacin, and that didn't change massively over this time period. And you can see that there was a big decline in the proportion of MRSA isolates that were resistant to ciprofloxacin from about 2006 to 2007 onwards. Now, at the same time, there was a call from the UK, from the NHS, to reduce fluoroquinolone use in part to tackle Clostridium difficile infections. It's something that we're looking into now, is actually getting GOSH wide antibiotic usage data, because all we had was antibiotic usage data from the patients positive for Staph aureus, to explore whether there was a general reduction in fluoroquinolone use across Great Ormond Street. Because fluoroquinolones are actually relatively rarely used in children. And so it's kind of it's great because it shows that we can reduce the levels of resistance to a key antibiotic in a key pathogen. But we yet to explain whether this was due to a wider decline in use of fluoroquinolones across the hospital. Now, at the patient level, we were interested in slightly different things. So we're interested in whether we could see this resistance gene movement, whether we could see evidence of phage. Well, we couldn't measure the phage, but whether we could see evidence of rapid gene movement within our patients. So we looked at patients that had multiple isolates recorded, and we found that about 1 to 4% of all patients at some point carried both MRSA and MSSA on the same day. And we found that the MRSA populations were much more diverse than MSSA, so you're more likely to have multiple antibiograms in your MRSA population, so multiple combinations of antibiotics. But the actual amount of diversity, so how different those two strains were, was about the same in MRSA and MSSA strains. We then thought we would look at patients that had different antibiograms on different days. And what we wanted to say here was, okay, so if a patient has susceptibility to an antibiotic and then they gain resistance to an antibiotic, is that because of selection? So is that because they've been exposed to antibiotics, or is that because of between patient transmission? So could we pull apart changes in resistance by looking at their antibiotic exposures to see if it was linked to selection, or by looking at whether patients in the same ward had that strain at the same time? So it's sort of a classic epidemiological analysis to see if we could find evidence of transmission. So what we did was we had patients who were positive for Staph aureus at time 1, and then within the same hospital stay, they had another sample that was positive for Staph aureus. And then we looked at whether they gained a resistance or whether they lost a resistance. And we looked at whether they had an antibiotic exposure between those two samples, with the hypothesis that it could be selection that leads to dominance of the resistance strain. It could be phage, just moving these resistance genes if they didn't have any antibiotics, then we had no, that was like our background hypothesis. Or it could be transmission. So we looked at whether the strain was present in another patient in the same ward at the same time. Now, when we did this, we found what to me was the most shocking of all the results in our paper. Yes, it was shocking that denominators changed massively over time. Yes, it was shocking, but great that ciprofloxacin resistance declined. But when I saw this data, it took me quite a long time to really believe what we were seeing and the implications for it. So we can see here the results of the proportion of changes in MRSA strains at the top and MSSA strains at the bottom. And the filled-in parts are the parts where that patient received the concomitant antibiotic between time 1 and time 2. And then the letters at the top tell you whether they gained resistance or lost resistance. So they moved from S to R, or they moved from R to S. Now, why this was shocking to me was, for example, for something like gentamicin, for any of these antibiotics shown across the bottom, what I would have expected to see was something like this. So maybe not as extreme, but basically, if I have a patient with a susceptible strain, they get exposed to an antibiotic, then they're much more likely to have a resistant strain at the next time point. that goes back, if you can remember, to those images that I showed you of the modeling of what my model is, what many models have of antibiotic resistance, that a patient with a susceptible strain is exposed during treatment or during prophylactic exposure, that causes a selection movement of a dominance of a population from being a susceptibly dominant population to being a resistant population, because they're exposed to the antibiotic, right? It kills off the susceptible strain, gives a selective advantage to the resistant one. So this is really unexpected result. The first paper that we published, and the link will be in the final slides, was really trying to describe what was happening at GOSH, looking at over time, really big picture analysis. And the analysis that we're doing now is really drilling into this, because I'd really love to hear your thoughts about why we get these patterns. You know, we haven't yet looked at whether patients got more than one antibiotic. So it's we just looked at resistance to antibiotic and antibiotic exposure on the very simple one-to-one correlation. We haven't looked at confounding because of antibiograms, so they might be resistant to another antibiotic that they also got in that time, and that plays within it. But I didn't look at timings, so we haven't looked at, for example, sample on day 1, antibiotic on day 2, second sample 2 weeks later, for example. So things that we have to look at to explore these patterns, but it's also quite worrying for me because this is routine surveillance data. So it's not all samples in a patient. It's probably representative of the dominant population. And so if that's not resistant after antibiotic exposure, then maybe resistance is there, but we're not finding it in the routine surveillance data. So we also, as I said, looked at transmission, and we found that about 50% of the events could be explained by potential between patient transmission. So we found the same antibiogram profile in the same patient, the same ward. But most of our resistant changes could not be explained by antibiotic exposure or nosocomial transmission. So if I was Quentin, then I would say, oh yeah, it's due to phage. We can find phage moving it. Okay, we don't have evidence for that, but something is happening. There is quite a lot of population dynamics movement. We found changes in our Staph aureus populations in our patients within a single hospital stay. And that's something that we need to look into more to see whether that matches up with what we understand about resistance gene dynamics. We also, in the previous one, we also haven't segregated it out by mutational versus gene-based resistance. Okay, so we couldn't find an explanation, but this is routine surveillance data, right? This isn't aimed at epidemiological analysis. This is aimed at treating patients, and so there is going to be a limit. There are going to be the fact that we found we could link so many was also a really good sign, and something we want to pursue going forward in terms of real clinical support tools. So what were we looking for? What can we do with routine data? Well, we can detect resistance changes. So this was new to our clinical colleagues. It was quite a slow decline over time. And so we can use that to retrospectively support them to understand what might have driven that. But we can also see the impact of personnel. These dramatic shifts in the denominator really affect our understanding of the routine surveillance data. And this is just one hospital, and this is happening because of personnel changes in different hospitals over time. I think it's a real worry for routine surveillance data. However, we could find some really interesting changes in resistance diversity. We could use this to, for example, model rates of resistance change with a little bit more work, and we could link a bit to transmission versus selection, but it was actually quite a bit hard. Now, I've emphasized here that this was data just from one hospital. It's actually just one hospital that had children in it. And when I was working on this paper, I was also doing a bit of work on my other favorite bug, which is Mycobacterium tuberculosis. And as part of that research, I was looking at trying to estimate the burden of drug resistant that is carried latently or asymptomatically in TB. And what we were finding there was that there were peaks in drug resistance at younger ages in kids for TB. And so we were sort of thinking about this and wondering, well, are MRSA rates higher in kids? Is this something we should worry about? And this got me to looking more widely in the literature and trying to see if there is a link between age and AMR. And so often there's a link between age and number of infections or worse outcomes of infections. But Actually, there's very little out there that quantifies how antimicrobial resistance prevalence infection varies by age and also by sex. And so this is the current project that I'm working on now, to try and do better on that, because this is really important. We think if there are, the ONIA report estimated in 2019, there would be around 700,000 deaths due to AMR, and around 1 in 11 of us would be over the age of 60. And this is set to dramatically increase. Both of these are going to dramatically increase by 2050. And there's potential for a huge collision here, in that there'll be more drug-resistant pathogens circulating, and our population will be older and therefore more vulnerable to bacterial infections, will receive more antibiotic exposure and have more contact with healthcare settings where often resistant pathogens are circulating. And so the collision of these two things, as I said, is what I'm trying to explore now and trying to quantify. Now, when I was trying to look for preliminary data on this, I did find one study from the OECD, which plotted the EARSnet data. So that's data from the European, from the ECDC. So that's routine surveillance data, mostly from bloodstream infections, a very small percentage from cerebral spinal fluid. And what they plotted here is a plot that I'm going to show you several variations of this, but on the x-axis is age, and the y-axis is the proportion of isolates with resistant bacteria. Now, remember I said earlier, routine surveillance data is mostly used to produce percentage of bacteria that are resistant to a certain antibiotic at the country level. And so that assumes that, resistance is flat across the board. There's no age effect. If I told you that we had data, for those of you that know a little bit about microbiology or about AMR, if I told you that I had data from these different drug and bacteria combinations, and if I told you, and TB is not on there, if I told you that one of them was MRSA, well, we know that MRSA circulates in hospital populations. We know that older individuals go to hospital a bit more. So maybe we might expect a trend like this for MRSA. And so what they actually found in the OECD report was there was huge variation in the levels of resistance prevalence in infection with age. And the yellow one there is MRSA, so we do find that there's an increase with age for that. But for others, for example, carbapenem resistance and Pseudomonas, it's a really, really different shape. And so this work I'm now pursuing with my postdoc, Naomi Waterlow, and what we did was we applied to the ECDC for the individual level patient data underneath this, and I have a fellowship from the UK Medical Research Council to really expand this and to quantify it in detail and to try and work out what's going on. So what you can see here, again, we're going to show lots of plots. On the x-axis is age and the y-axis is AMR. And the different colors of the different drug families, so aminoglycosides, and then the beta-lactams split by their wear grouping, and then the different types of lines of the different sexes. And here we just use the male-female as recorded in the ECDC data. Now, again, the y-axis is percentage of isolates, is actually proportion. So proportion of isolates tested that are resistant, and then each of the different columns, the panels that you'll see, the different antibiotics. And the rows will be the different types of bacteria. So I'll just flash it up here because I think it's really useful to get an overview of what we've got, because at the top, you've got your gram negatives and at the bottom of your gram positives. Now, hopefully, the first thing to see is that there are lots of different patterns, and it's not common, as was in that OECD report, it's not that there's a single pattern for how resistance prevalence infection varies by age. If I focus on few key examples, then I'm highlighting MRSA down in the bottom there. E. coli is across there at the top, obviously one of the biggest drivers of bloodstream infections, and Pseudomonas aeruginosa has this sort of N-shaped pattern going on there. Now, if we think about looking for certain patterns, it's quite hard to pin down patterns, but if you look at a certain drug, for example, if we go to the fluoroquinolones, the sort of lighty yellow colors, or even something like the macrolides, the dark blues, you can see that within a color, the patterns aren't the same. So the trend in resistance with age isn't the same. So there's more variation between drugs than within bacteria. So there are some bacteria that have patterns of sort of multi-drug resistant clones. For example, look at that pattern across age for Pseudomonas aeruginosa or Acinetobacter in the top left there. Now, what was really interesting was that despite the fact that that women are actually exposed a lot more to antibiotics, sex had very little impact. A bit more for the gram negatives, for example, you can see some differences here, and for Pseudomonas, for Acinetobacter at the younger ages, but it wasn't as big as an impact. We're expecting to see a lot more differences by sex. Now, this is the key thing to emphasize here is this percentage of isolates tested that are resistant. If we look at incidence, we know that incidence of bloodstream infections dramatically increases with age. It's higher in males, especially at older ages. But we did find some interesting patterns that women have more bloodstream infections due to E. coli between the ages of 20 and 40. We're not the first people to show this. It's in some UK, the S-Power reports, for example, but I'm yet to find a good evidence-based reason for this. I mean, again, I'm sure you can think of them, UTI, childbirth, for example, but actually linking that through to the causative mechanisms is something that we want to do. So if we go back to that graph, then what we tried to do, so this is at the European level, it's data from 2015 to 2019 pulled together for these bloodstream isolates. And this raises just lots of questions about AMR epidemiology, what the drivers are, why it's so different. And the next thing we wanted to do was try and understand what was different at the country and subnational level. So we did that, we did that for all of the countries we had, 28 European countries. And we fitted multi-level regression, Bayesian multi-level regression models to this to try and pull it apart. I'm just going to show you the results of that for two key examples. So the aminopenicillins here and for methicillin. So aminopenicillin resistance in E. coli, as I said, is a big problem in drug resistance, and MRSA because it had this clear increase with age. Now, what we did was we fitted our multi-level regression models to this, and what you can see here, the results of the model output for 3 example countries for these 2 drug-bug combinations. The top is for females and the bottom for males. Now, you can see that the trends are country-specific, so variation in the shape by country. There's also a big range between labs, so a lot of subnational variation. So each lab, we had that in the data, is shown as a different line here. And we fitted our multi-level regressions at the level of the lab. And so you can see big variation in the prevalence of resistance. And then we tried to come up with a simplifying index. So we looked at the difference in model predicted resistance between the young and the old. And this is pretty robust to what we call young and what we call old. But here you're showing for the difference between model predicted levels of resistance at age 100 to age 1. And here you can see for these example bug drugs, it's mostly positive for MRSA, so mostly increase for age, a decrease for APEC, and mostly higher in males for MRSA and lower in males for aminopenicillin resistance in E. coli or APEC. So I hope what I've showed you there is that there are big bug drug differences in how AMR varies by age and by sex. They're also country specific, and we really need to exploit these patterns to understand and intervene now in AMR. It's opened a big box of questions about the epidemiology of AMR. And we need to use it to understand future burden predictions, and because these fundamentally underpin what we can say about interventions against AMR. So what we're doing now is building mathematical models that try to account for some of these reasons, and I'm sure, and I would love to hear your reasons why we see these differences, and things like antibiotic prescribing differences, immunosenescence changes, behavioral contexts, and we're trying to put these into models to test and explore and fit to this data to say which of these things really matter, and therefore where can we intervene. So I'm going to wrap up there very quickly, just talk through some take homes. I hope that you're convinced that listening to a mathematical modeler and someone who thinks about how to build the dynamics and where patients flow and why this matters and movement of resistance genes can act as a campfire and make you think about things a bit differently. And hopefully you're going to go home and write down your own models and try and understand how things work. As I said, I'm a convert to routine surveillance data because that's the data we have, and that's what we're going to have globally if we can get infrastructure and mechanisms in place. But we need to challenge it. We need to be really careful that we're combining it with our knowledge of what antibiotics are being prescribed, who's being sampled, to really say what do we know and what don't we know about AMR prevalence. And particularly to think about how it might vary by different subpopulations, for example, by even simple demographic indicators that we don't quite understand yet, like age and sex. So lots of people to thank, in particular, obviously the patients, clinical microbiologists and clinicians involved in both the GOSH work and also the EASNet data collection, and my collaborators, in particular Quentin, and obviously the rest of my group. There are some papers that you can find, and the demographic work is up on a preprint, and the GOSH work is already out there, and the TB drug-resistant patterns is also out there as well. Very happy to take any questions by email. I think Wendy's also happy to take questions by email, and do sign up to our AMR Centre newsletter if you want to hear more, and do join our webinars. But I'm going to finish there and let Wendy lead the Q&A.
Thank you so much, Gwen. It's a very interesting, very thorough presentation. So we'll get into the discussion and the Q&A period. So just a reminder again, if you have any questions for Gwen, please leave them in the Q&A. We do have some questions already there, so I will be going through those. So first, Vince would like to know, what role does wastewater environmental surveillance play in AMR modeling? Could established ES programs, such as for polio, be leveraged to collect more data?
Great, yeah, thanks, Vince, for this question. We're trying to organise a wastewater surveillance AMR Centre day at the London School, so do watch out for that if you're interested. But yeah, it's a question that we're trying to work out now at the moment. As you say, huge advances because of COVID and polio in this detection. I think what we're missing at the moment is an understanding of how resistance prevalence in wastewater correlates with resistance prevalence in infection, and how much of the changes in the bacterial population could be due to antibiotic exposure in the wastewater and in the environment. And so I think the studies that we need now are we're measuring resistance in patients in hospitals, measuring it in upstream, downstream wastewater sources, and saying, does it correlate? And then also trying to understand what are the questions we want to ask, because again, we probably actually don't want to measure hospitals. We probably want to do it in community settings where we're not sampling from patients. We probably want to do it in those kind of settings, and there it could be really useful to understand colonization, as long as someone can do that correlation, like how much selection is happening outside of patients, and how much of it is like an indicator of environmental pressures, and how much of it is an indicator of human or animal disease issues. Yeah,
perfect. Thank you so much. So next one is from Roxanne. So just from an AMR prevention standpoint, what is your view on the use of antibiotics as a prevention strategy for bacterial SDBBIs? For example, doxycycline post-exposure prophylaxis.
Yes, I don't know what STBBIs is. That sexually transmitted bacterial?
Yes.
Okay.
Yes.
Yeah. So again, this is an interesting question. Antibiotic it's something that came up when we were thinking about how worrying is COVID for AMR, because huge amount of patients being given a lot of antibiotics, and so kind of sort of similar, like huge amount of patients giving antibiotics potentially for not a bacterial infection or as a prophylaxis. I think we don't have a good idea of what that does to your microbiome, your nasal, your vaginal, your gut microbiome. So the knock-on, potentially bad effects of long-term gut microbiome changes, which might prevent that one infection. I think we don't know about that. I think if the trials are showing that it has a good preventive strategy, these are infections where we are struggling to get patients in the door. We are having high loss of follow-up and everything. So it's going to have to be a trade-off. I think what worries me a bit about some of these things, like the mass drug administration in low-income countries, to sort of to account for other problems. As long as we can measure the knock-on microbiome effects and understand them and see that as a potential con, then obviously the pros are easy to measure, and it's working out that difficult balance between the acute direct impact of that exposure and the potential long-term offsetting problems that I would worry about. But yeah, obviously, everything has a pro and a con, and it's the acute benefit of that might be quite huge. I'm not an expert on STIs, so yeah.
Perfect. Thank you so much. Next question is from Manish. Thank you for the great presentation. Due to globalization and ease with worldwide travel, we get more patients with antibiotic-resistant organisms admitted to our facility with travel history, resulting in transmission of these organisms within the hospital setting. The AMR reported by the microbiology lab is substandardized in low to medium income countries due to lack of standardization of microlab and lack of knowledge regarding the current guideline from CLSI or EU CAST. So what strategies can be used to overcome these challenges to curtail AMR? What are some low-hanging fruits?
Yeah, thank you.
Longer question.
So travel and the importance of travel at different ages, it comes up a lot in terms of trying to explain some of those patterns. So in Europe, we don't have big what's sort of the word, in-house problems with carbapenem resistance. Often there's a worry that these are imported from travel, and so that's definitely something that we're going to go forward as one of the reasons for potentially explaining some of these shapes, that they are importations and travel associated. I think I don't know if you know about the Fleming Fund, but that's a great initiative led by UK government and partners that is trying to basically improve routine surveillance and say, we need more microbiology labs, we need more sampling, we need more and better antibiotic prescribing based on microbiology results. It's difficult to say if that's a low-hanging fruit because that involves a huge amount of investment, not only in infrastructure, but in patients. I think there's also some really great work that's being done to just report microbiology data and resistance prevalence back to patients, back to decision makers at the local level, to say to them, this is your AMR problem, this is what you need to do. And this, I think that's probably the lowest hanging fruit, is sort of open access, simple data analysis tools that empower low-income countries to do better with their own data and understand their own problem and go forward from that. And that might also be an access problem, that we don't have access to certain antibiotic. We shouldn't be worrying about resistance because we're not sampling. I mean, a lot of these things from my interaction, clinicians know about them and are aware about them, and they do need those better labs. And they do want to use the right antibiotic. They know they're not sampling everybody. They know they've got a biased resistance profile, but supporting them to do better with their own data would really help to sort of empower everybody to get these things moving, which is why I think what the Fleming Fund is doing really great in terms of just raising, let's just do better routine, let's do better at that, and use that to inform local decision making. I'm not sure it's a low-hanging fruit, but it's something we should be doing.
It's difficult. So next question. Duration of exposure to antibiotics may also play a role, and it may not just be age. So should that be considered separately? And that's from Nalini.
Yeah, that's a great question as well about duration and then also frequency, right? So there's some great work coming out of the US that's showing that 10% of patients that get prescribed antibiotics get prescribed 40 times more than the rest of the 90%. I'm making the numbers up there, but I definitely think we don't know enough about whether it's a binary on-off. One day of antibiotics is as bad as 3. It's probably going to vary by the type of antibiotic. It's going to vary by the patient's PK/PD exposure, ability to remove that drug from their system. And it's something I mean, to be honest, we haven't thought about that because I don't think we'll get the data very easily. We're struggling to get, pure, total antibiotic usage by age. We're struggling to get, it's very rare to find age and sex disaggregated, openly available data. But I think, Nalini, it's not my research, not my literature to look at, but there are some research in the US. If you look at things like frequency versus duration, there is some stuff out there about the importance for AMR, yeah.
Thank you. So running a little short on time, but really quickly, Yahaya is just wondering if the OECID data.
So the OCD data is the report that I mentioned is it's $1 more, which I think was published in 2018 or something, that has that in there. So the ECDC data I can't share, but you can apply to ECDC through the TESI system for application. You have to write a protocol and ask for permission for that. They might, it depends where you're located, whether they'll share it with you as well, because it's European data. But yeah, contact them through TESI. I think it's something like data.access at ECDC, but if you Google EARSnet and TESI, then it should come up.
Okay.
or any other web searcher.
Okay, so we are at the end of this event. So thank you again, Gwen, for your presentation today, and to everyone who was able to join us for this webinar. I know we weren't able to get through all of the questions. So if you would like to have those questions answered, please send them to us at nccid@umanitoba.ca. And we also have a podcast episode with Gwen where she discussed some other aspects of her work and about AMR modeling in general. So please tune into that as well if you're interested. You can find the Mod4PH podcasts under the webcasts tab on the NCCID website. And that's also where the recording of this webinar and the slides will be soon available. So yes, finally, take a moment, please, to fill out the short evaluation, which will be sent out immediately after this event. And once again, thank you so much, Gwen, for joining us today. And I hope everyone enjoys the rest of their day. Thank you.
Thank you.